problem, as you know, is that malaria affects hundreds of millions of people every year and kills over 500,000 children. In the 1950s, there was lots of optimism about the idea that malaria might be eradicated. And a lot of that optimism stemmed from the wonderful drugs and insecticides that were around that time. At that point, chloroquine had been discovered as to be a highly effective, cheap, safe anti-malarial drug. And DDT had proved to be a highly effective insecticide. And there was a global malaria eradication campaign mounted in the 50s and 60s involving the World Health Organization and many governments around the world, and huge amounts of money were put into it, a massive logistical exercise. But by the 90s, there was gloom and despair because it now seemed impossible to achieve malaria eradication. And those of you who work in tropical medicine will know that's an extraordinary, the reasons for that are very complex. A lot of them are to do with practicalities, logistics, politics, and so on. But a crucial factor was that now the parasites had become resistant to the antimalarial drugs and the mosquitoes had become resistant to the insecticides. And on this map, which you can find on the WARN website, what you can see is how resistance to frontline antimalarial drugs has evolved since the late 50s to the present day. And what you can see is the spread of resistance to chloroquine, to sulfadoxine pyrimethamine, the former frontline drugs, and in purple to both of those. So really, those really are no longer effective in tackling this problem. Now, as you probably know, we have a wonderful new drug, artemisinin, which is now deployed in combination with other drugs to prevent the spread of resistance. But unfortunately, that has not been successful, and the reasons for that are probably extremely complex and poorly understood. I'm going to speak about some of the underlying evolutionary reasons for that, but there are probably many other factors as well. But anyway, artemisinin resistance has now emerged and is spreading through Southeast Asia. And at the same time, the mosquitoes are developing resistance to the strategies we're using against them. They're developing resistance to insecticides and to bed nets, and they're developing that resistance. It's all genetic, it's all evolutionary. That resistance in the mosquitoes is complex. It's partly about chemical resistance to the drugs, to the insecticides, sorry, but it's also about behavioural shifts, so that they tend to bite potentially outdoors instead of indoors, and that changes the efficacy of bed nets. So there are complex things going on. And put very simply, when we attack Plasmodium with drugs and Anopheles with insecticides, the parasites and mosquitoes fight back. And that's just a general law. Now, I want to talk a little bit in my talk about some of the underlying genetics and evolutionary mechanisms of that. And for those who aren't into this field, you may want to switch off for the next 10 or 15 minutes, take a break or something, but I'll try to explain it in such a way that it'll be accessible even if you're not a molecular biologist or a geneticist. And a crucial question is, how often do parasites or mosquitoes develop new mutations that might give rise to drug resistance? And I'm going to focus almost all of my talk on the parasites, although I will speak briefly at the end about the mosquitoes. But some of the same general principles hold for both. Let me begin by saying that in contrast to what David was talking about in the previous talk, and thanks for the introduction, David, because it was really helpful, but whereas David was talking about potentially viruses and bacteria that may have hundreds or thousands of variants, sometimes more than that, in the case of the parasite genome, which is 23 million bases long, it's got millions of polymorphisms, millions of positions in the genome that are varying. And in the case of the mosquito, which is 250 million bases genome, we've got potentially hundreds of millions of variants. We're finding a variant every 4 or 5 bases in the mosquito. So these are vastly complex organisms approaching the complexity of humans. In fact, in the case of mosquitoes, maybe more genetically complex than humans. And that means, in practical terms, that just as we look around this room, and you all look pretty different to me, and I guess you all behave differently, and you've got many other differences. The same holds true in parasite and mosquito populations. So it's a complex sort of set of problems we're dealing with. And just as in human populations, for most things that interest us, there isn't a single gene that controls it, but genetics clearly is involved. The same holds true for parasites and mosquitoes. So we're talking here about a vastly complex problem and really big data. Anyway, to come back to the simple things, how often does the parasite genome mutate? And to get at that problem, actually, you've got to do experiments in the laboratory, and a beautiful set of experiments done by Will Hamilton and Antoine Classons and Mihir Gekri in our lab at Sanger over the last few years has involved so-called mutation accumulation experiments, where parasites are grown through multiple generations and continually cloned and recloned, and by sequencing the clones that arise from that process, you can work out how often new mutations occur. And put very simply, we can estimate that the P. falciparum parasites would generate a new mutation at every position in the genome, all of the 23 million positions, roughly every couple of days in a single person who had malaria fever. So if you want to sort of think about the mechanisms by which, say, drug resistance emerges, it doesn't involve the Wizard of Oz coming down and anointing, a particular parasite in the middle of nowhere. It's actually the mutational process is going on all the time in a single infected person. And actually, parasites are fascinating because as well as that process of mutation, they're shuffling things around, and actually they're particularly shuffling around the genes that encode the variable antigens, and they're generating antigenic diversity in terms of millions of new forms every day or two. So basically, mutations are out there. So what interests us then is how evolutionary processes, an epidemiological process, act on those mutations and make them emerge in the population and spread. They're happening in the odd parasite. So if you've got 1 billion parasites inside you, there'll be a particular interesting and nasty mutation lurking in one of them. And the question is that one going to make it through to a mosquito and infect another person? And if so, is it going to spread from that person to other people and end up going through the population? Or will it just vanish? So to get at these questions, we have to start to go to real populations. And the Malaria Genomik network, one of the main things we've been trying to do, getting people to share data, develop common standards for data sharing. But the crucial purpose of all that is to construct an observatory as to what's going on in the real world. And we have a project called the P. falciparum Community Project, where we're working with investigators in over 20 malaria countries and sampling parasites. Working with them, they're often doing very interesting studies on those parasites locally, but we take those samples, we sequence the genomes, and we give the data back to our partners. They may use it for their own studies, but we also put the data together at a high level to construct a view of what's going on. And here's an example of a particular study we're working with. You can find it on the web. Which is the TRACK study. It's the Tracking Resistance to Artemisinin in collaboration, and I'll be talking a bit more about it later, which is sampling from multiple sites across the Mekong region of Southeast Asia. So that's the first part, sampling from the population and attaching those samples to good phenotypic data and genome sequencing data. The next thing we have to do is to characterize the diversity we see, and I could speak for the rest of the day about the challenges of sequencing parasites and analyzing the data, because it's a very complex genome. I'm not going to do that, though. I'll just simply say we do all that sort of stuff, and that's what many members of our team and our colleagues are working on. But we do put the data on the web, and I'll talk a little bit at the end about how you can access this, but put very simply, if you Google on Malaria Gen and click the buttons at the top right-hand side of the page, you'll hopefully be able to navigate to maps like this, which show you, in this case, pointing. I'll just wave my arms, which basically, in this case, show you going across the horizontal axis, a region of the parasite genome, a small region of the genome. You're seeing at the top there a particular gene. You can see on those triangles there are particular variant positions in the genomes, so-called SNPs or single nucleotide polymorphisms. And what you're seeing on the vertical axis is different samples from the population, and what we're seeing, the colour is whether they, in blue, resemble the parasite reference genome, or in red, whether they are different from the parasite reference genome, variants. And what we're seeing in black is bits of the genome that are missing. In this case, it's because there's a large deletion in that part of the genome in some samples. And you can navigate across the genome and find this type of data. Now, the next thing we want to do is to characterize the diversity of the population. And although we're talking about millions of variant positions, we can do some rather simple tests. We can reduce the complexity of that data and ask, how similar is one parasite to another parasite? And as I said earlier, basically all parasites are different from each other. But often, that was a very broad statement, but if you sample parasites from one person, you'd probably find they're different from the parasites in another person. And what we can see in this so-called neighbour joining tree is data on over 3,000 individuals from a study that we're writing up at the moment, this PFAS-CIPRAM community project, and these data representations are by Oliver Miotto and Roberto Amato. And what we can see around the outside of this diagram, each line on the diagram represents a single sample. And what we're doing is the algorithm here is grouping samples by their similarity, and you can see they're all different, but you can see they cluster in rational ways. So on the left-hand side, we see samples from Africa, and on the right-hand side, samples from Asia, and then samples from Oceania and South America live somewhere in between those. And we can see that in Africa, we could divide samples up from those in West and Central and East Africa. And in the case of Southeast Asia, we can see there's a substantial split between the samples in the eastern part of Southeast Asia, Cambodia, Vietnam, from Western Southeast Asia, Myanmar, and the western border of Thailand. So in brief, parasites are different from each other, but parasites that live near each other are more similar to each other than parasites that live a long way from each other. And in that respect, they're quite similar to human beings. And actually, that's not accidental, because most of them live inside human beings. So there's a connection there. Human beings are the major vectors of malaria, just in case you were unclear about the way it works. Okay. So now we can reduce the data still further. And this is another sort of way of compressing all that data. It's a so-called principal components analysis. And again, it allows us to look at similarity between samples, but in a slightly different representation. And when we did this analysis some years ago, we were totally puzzled by it, because what we could see is a group of samples we collected in West Africa, and that included various different parts of West Africa: Mali, Burkina Faso, Gambia, Ghana. They all clustered together. And similarly, when we looked across Southeast Asia, across Cambodia, Vietnam, and parts of Thailand, Myanmar, we found they clustered together, but there was a group of samples that seemed to sort of split away from those clusters, and they were all turned out to be from Western Cambodia. And at first we thought that was must be an artifact, but we investigated this phenomenon. We went into it in some detail, and you can read all about it in a paper by Miotto et al. In Nature Genetics a couple of years ago. And the long and the short of it is that these odd parasites on this tree are newly emerging strains of artemisinin-resistant PFAS-CIPRAM. We could see they were quite different from the others. And I'm not going to go into all the details of that. It's a rather complex population genetic phenomenon. These were multiple independent founder effects. We could see there were multiple strains of artemisinin-resistant PFAS-CIPRAM spreading through Western Cambodia. And that's very interesting, because Western Cambodia that region is where resistance has emerged from on multiple occasions. So in the 1950s, that's where chloroquine resistance started and then spread around the world. And in the 1970s, resistance to Fansidar, sulfadoxine pyrimethamine, and now artemisinin resistance. Now, as I said at the beginning, many of these mutations are happening all the time, and actually there have been other independent mutations of chloroquine and Fansitar resistance. So I don't want you to think the Wizard of Oz only lives in Western Cambodia, but there is something special about the mutational process going on there that seems to breed a particularly nasty strain that's able to spread around the world. So this is extremely important that artemisinin resistance is occurring there right now. At the end of 2013, researchers at the Institut Pasteur in Paris and in Cambodia published a wonderful piece of work which stemmed from experimental studies they'd been doing in the lab for several years, where they'd been breeding artificially a strain of P. falciparum that was artemisinin resistant. They'd been feeding it with artemisinin and probably topping up with a little bit of red wine and so on. And they got a mutant strain that was resistant to artemisinin. And then they sequenced that and they found a number of interesting mutations. And then they explored those mutations in samples they got from Cambodia, and they matched those data up. A wonderful piece of detective work. And they basically provide fairly compelling evidence that mutations in a particular gene we'd never sort of thought about before, called Kelch13, a Kelch-like gene on P. falciparum chromosome 13, those mutations seem to be very strongly associated with artemisinin resistance. And subsequently, and I should just point out that this was not just mutations in that gene, that was mutations in a particular region of the gene, a so-called propeller domain, a very structurally conserved domain of the gene. And subsequently, there's been a lot of work on this by multiple groups, experimental work and epidemiological work, and now it's quite clear that these mutations cause artemisinin resistance in Southeast Asia. But the remarkable thing, it's not a single mutation that's doing it. There are multiple independent mutations, and it's now clear from a number of studies that there's at least 20 different mutations circulating in Southeast Asia, independently arisen, which occurred in the propeller region of this gene, and they're all independently associated with slow parasite clearance when a patient is receiving artemisinin. And in this graph, what we're seeing on the horizontal axis is different mutations in this gene. On the left-hand side, we're seeing mutations outside the propeller domain, and on the right-hand side, we're seeing mutations in the propeller domain. And on the vertical axis, we're seeing the parasite clearance half-life in patients receiving artemisinin, how quickly the parasites were cleared. And you can see the clearance times are significantly longer in those with propeller domain mutations. And a very, very detailed study of this was done by the TRACK network, published in New England Journal last year. And recently we've published another analysis of that, more detailed, to do with the underlying genetic architecture in Nature Genetics. And I'll say a word now about that more detailed study. So what we wanted to understand, working with the TRACK network, is it just the Kelch mutations that are causing resistance, or are there other genes involved? And in particular, can we find anything out by looking across the whole genome to get any clues as to why these mutations are occurring in this particular part of the world? And here's the GWAS so-called Manhattan plot. Does anyone here know about genome-wide association studies? Not a big uptake on that question, so I will skip over the details. But suffice to say that what you're seeing on the horizontal axis here is the whole parasite genome spread out in different colours. You're seeing the different chromosomes, and on the vertical axis we're seeing the p-value for the question of, is there a genetic association between that part of the genome and the phenotype of artemisinin resistance, in this case as determined by slow parasite clearance. And we can see some astronomical p-values, 10 to the minus 20 and more. So we've got some very strong hits. And there are two take-home messages from this slide. The first is that unquestionably Kelch mutations are the strongest associations with artemisinin resistance, but also there are a number of other loci in the genome with extremely strong statistical evidence of an association. So there are multiple genes involved. But statistically, there's a fascinating phenomenon. If we now condition on the Kelch mutations, in other words, if we take that, if we allow for that, the question is what happens about all the other associations? Are they contributing independently of the Kelch mutations, or are they involved with it? And the answer is, if you take the Kelch out of it, the other associations, if you account for the Kelch, the other associations are no longer significant. So what's happening is the other mutations don't appear to be contributing to the phenotype, but they hang around in some way with the Kelch mutations. Did that make sense? Let me put it another way, because it may become more clear if I represent it like this. The conclusion we draw from this is that it's the Kelch mutations, these independent Kelch mutations, that are causing the resistance. But these other loci in the genome, these other genetic variants, are strong markers for the sorts of parasites that develop resistance mutations. They're predisposing factors for the emergence of resistance mutations. So if I link this in with what I said earlier on, Kelch mutations are happening all the time, but if they happen in parasites with this particular genetic background, they're much more likely to take root. I mean, I guess that concept must be familiar to anyone who does gardening. You know, you can seed things all over the place, but they take off in some places, but not others. That may not be the best analogy, because I'm not a gardener, but you know, you get the general point. That on this genetic background, which happens to be particularly common in Southeast Asia, these particular mutations take root. And actually, what we can see here on this representation is a map of mutation frequency. Now, if you looked at the map on the left-hand side, this could well be a map of just the artemisinin resistance phenotype. In red, effectively, this is places where there's a lot of resistance, and you can see it's focused on Western Cambodia, and in white, or cream, are areas where there's little or no resistance. But actually, I'm not showing you the phenotype here. That's what it would look like, but what I'm actually showing you here is the frequency of these Kelch mutations. And I'm not showing you the frequency of one mutation, I'm showing you the aggregated frequency of all of these independent mutations. And on the right-hand side, you can see maps that look very similar, and this is mapping the frequency of these variants that predispose to the emergence of mutations. So all very fascinating. How does it work? No idea. Now, the question is, has artemisinin resistance spread to Africa? So now that we know Kelch mutations cause resistance, it should be dead easy. We go out and sequence genomes or sequence the gene and work out, is there a mutation, so there's going to be resistance. And when we count the number of different positions in the genome we see with a Kelch mutation, we find roughly the same number in Africa to Southeast Asia. So that sounds pretty scary. So have they spread from Southeast Asia to Africa, and are they now carrying artemisinin resistance there? So the first thing we've discovered, and we know this because we're sequencing the whole genome and we can do some detective work about this, it's very clear that the Kelch mutations that we find in Africa come from Africa. They haven't been imported from Southeast Asia. And we can do that by studying the rest of the genome and ask the question is, does the whole genome, or does that particular region of the genome looks like it comes from Africa or Southeast Asia? So these are homegrown mutations. And the other thing we can work out, and I'm not going to go into the statistics of this great length, but we can work out that the ones that are seen in Africa show no evidence of being under evolutionary selection. Firstly, because they're very low frequency, they're much lower frequency than the ones we find in Southeast Asia, but also because when we look at the gene in general, we find various features that tell us it looks like it's just doing its normal business of random mutation that I talked about earlier. So, for example, we find no excess of non-synonymous SNPs over synonymous SNPs. A non-synonymous variation is one that changes the structure of a protein. A synonymous mutation is a mutation that changes the DNA sequence, but doesn't change the protein, so it has no effect. And we find no excess of that ratio in Africa, whereas in Southeast Asia, it's way off scale. So clearly, these Kelch mutations are under massive pressure in Africa and so being selected, Southeast Asia, sorry, but not in Africa. Now, we could have a very interesting discussion about the reasons for that, and we don't know. And anyone who works on malaria will realise that probably a lot of the reason is there's less selective pressure, less drug pressure in Africa for multiple reasons, partly to do with patterns of drug usage, but also because in a highly endemic setting, quite a lot of the parasites are sitting around in asymptomatic individuals and are not getting challenged by drug treatment. So that's probably part of the reason. But we think there are probably quite a few other things going on, including possibly this question of a genetic background. And it comes down to questions of biological fitness. So mutations happen, but whether they spread depends on whether they are good, how well they do against their peers in the long run. So a parasite may do extremely well against its peers in a particular person who's receiving artemisinin treatment, and it'll zip ahead of the other parasites and it'll be the one to make it through to the mosquito. But when it gets into the mosquito, it may find it rather difficult to compete with its peers. In fact, it may be a disadvantage getting through the mosquito because that mutation wasn't good for it in the mosquito. And when it gets into the next person who's not receiving artemisinin, it lags even further behind. Yet it's a long-distance race the parasite is going through. It has to get through not just that person receiving the drug, but it has to get through the mosquito and the next people beyond that. So that aggregating that biological fitness, that whole equation is probably different in different ecological and epidemiological settings, and it probably is different for parasites on different genetic backgrounds. And again, the analogy with humans is you can probably sort of see the issues here. People are sometimes good at one thing, but actually it makes them not so good at something else. Trying to understand those processes is fundamental. Now, sorry for that long excursion into the underlying evolutionary biology, but the point is that if we're going to try to develop the sort of methods that David was talking about, usefully using genetics to control disease, we've got a pretty complex job on our hands for malaria because there's just a lot more stuff going on there. And we're not in the position right now to just come up with a simple set of tools of the sort that David has said that's going to help us to manage, control and eliminate malaria. But we can see that the direction of travel is that with big data and with appropriate technologies and with appropriate processes, it will be useful in the future. And I want to sort of explain why it's so important to have this sort of information. So imagine that now, coming to the real world, that you're responsible for eliminating malaria from a particular region of the world. It's very, very exciting. It's the job you've always wanted to do. You've been given $10 million. You map out the lie of the land. You identify all the transmission hotspots. You rev up the Land Rovers and you send out the field workers and go. So this is how you start. You've mapped out the region, and what we can see in blue dots here are the transmission hotspots, and in each of those transmission hotspots, the parasites are sensitive to the drugs that you're using. So you get going, and halfway through your $10 million spend, this is what it looks like. You've done a pretty good job. In most places, you've reduced the level of transmission, gone from a dark blue to a light blue. But in one place, the parasites at that point, because of the pressure you've been putting on them, have become resistant. So now you've got a couple more years to go, $5 million to spend, so you get going, and now two things can happen. Either you get rid of all the parasites, or you turn them all into resistant parasites. And now most people will probably want to end up with the top right scenario as opposed to the bottom right scenario. And the reason they get into the bottom right scenario is not because they wanted to get there, but because they didn't know what was going on in the middle. They didn't know about that resistant hotspot. If they did know about it, what they might do, even if the drugs were not effective, is put all of their resources into other methods of malaria control, vector control, for example, and other measures that would have taken out that central hotspot. You don't have to use artemisinin for that. There are other ways. So it's a question of resource allocation. At this point, the most important thing in the middle of this diagram, the most important thing that the person can do is nuke that particular hotspot. So to translate this to public health applications, we need real-time data for malaria control teams to allow them to navigate that. But equally importantly, this is not just about real-time data to support teams on the ground. It's about real-time data that is needed for strategic planning. Because if we're allocating now resources to a particular strategy, be it mass drug administration or test and treat or to vector control, whatever, we want to be able to model what the likely impact of that is. And understanding how the parasite population is responding is crucial for that process. And genetics is going to be useful for that. And so right now we have a little bit of a problem. I've explained to you it's a complex, genetics is of parasites and vectors is complex. And right now we're in a cycle where it's taking potentially years to get from the point where resistance emerges to understanding what are the markers we can use to track it. And typically we're producing the information about those markers in scientific publications, which in our case can take years of peer review. I know some people are faster in getting their papers through the reviewers. And it comes out into the real world much too late to be useful. And everything needs to be speeded up. So that really we're finding these changes in real time and we're putting the data out there in real time. So in terms of supplying information to teams on the ground, we need to be in a position where where control teams are routinely collecting dried blood spots that are being pushed through into high-throughput genotyping labs. All the statistical and computational algorithms are being worked on them, and maps are being fed back to the teams on the ground as to where resistance is taking off and spreading, and that could all be centrally controlled through some sort of mobile application of the sort that David was showing you. So that's the sort of place we want to be. So 50 years on, what we'd like to do is improve on the situation in the original global malaria eradication campaign. This is a picture of one of the surveillance labs, and you can see loads of glass slides and loads of people reading them, and it required a lot of training, and sometimes they ran behind and slides got lost and data got stuck in the bottom drawers of people, and it was a real problem. But actually now this could be moved on to a much more effective mobile surveillance system with open access data in real time. Now, how am I doing for time?
You've got plenty. About 20 minutes.
Oh, right. Okay. That was the central part of my talk about parasite drug resistance, its evolution, and where I think we need to be going. There are a lot of data out there that I haven't talked about, and in the closing minutes, I'm just going to talk a little bit about some of the other data that you can get online. Now, if you're not into genomics, again, you can probably switch off here, because what I want to say is that we're generating terabytes of data, and the question is how we make that all accessible, that people can play with it. As I've said, it's a very complex scientific problem, and we want many people to be looking at the data, to thinking about it, developing their own tools and algorithms, and thinking about different ways of applying the data. So in Malaria Gen, what we've tried to do is develop a set of resources, a set of software that allow people to look at data as we generate it. And I've done some slides for this, but if I've got a few moments, I'll try to do this live. So let's see how this works. Okay, so if you Google on Malaria Gen, what you'll find on the front page, or the top right of the front page, you'll find a number of web applications you can click on, and I hope that if you click on any of them, you can sort of guide yourself around. We're about to produce more online tutorial material, but we hope that most people who are savvy at learning how to use web apps can sort of navigate, can find some useful things just by clicking at it. And these will work. They work best on Chrome, but they'll work in any web browser, and they work pretty well on an iPad. They're a bit too big physically to be visible on an iPhone or a smartphone, but on a tablet they usually work pretty well. So if we now went to this one, which is the AG 1000 genomes application, what we've got in this application here is data on almost 800 samples of Anopheles gambiae from across Africa, working in a consortium that involves many of the major groups working in Anopheles gambiae population genetics. And so this is a consortium led from the Sanger Institute, but it involves groups around the world. And we've now released whole genome sequencing data, as I said, on almost 800 mosquito isolates from different locations across Africa. I mentioned earlier that the mosquito genome is highly polymorphic. Its diversity is at least an order of magnitude greater than humans, so that on average in human beings, if I took any two human genomes, example, I took the genome that your mother gave you and the genome that your father gave you, on average, I'd expect to see about one difference every 1,000 bases. Whereas for mosquitoes, it's more like one difference every 100 bases, or even more than that. So vastly polymorphic. And if we look across the whole population, we find about one difference every 4 or 5 bases. So with this vast amount of data, we need tools to navigate that. And one thing we can do is we can look at a chromosome. So this is the Anopheles gambiae 2L chromosome, and across the top, going horizontally, what we see the chromosome, and it's 50 million bases of DNA sequence that we're reviewing here. And what we can do on the browser is we can sort of zoom in, and as we zoom in, we start in a minute to see, if we watch the top panel, we're starting to see things there, and those things there you can see in the top panel appearing, they're things called genes. They're the things that encode proteins. So we zoom in further, and we're looking at genes, and now we're looking at individual genes, and we keep on going, and we keep stretching and stretching and stretching, and now in a minute what we're going to start to see appear below that are little triangles, and those triangles are mutations, they're variations. And what we're seeing down at the bottom there are samples, and in those samples we're seeing in blue whether that mutation looked like, whether that individual mosquito looked like the reference sequence, or in red whether they look completely different from the reference, or in green whether they were heterozygote, where half looked like the reference genome and the other half similar, half different from the reference, heterozygote. And what we can see in the areas of white are areas where our algorithms right now are not very good at calling that particular region of the genome. It's relatively complex. What we can do if we go back to the start page is find a whole range of different things you can do with the data. So I just did one thing, which is look at the genome and stretch it, but we could go in and we could look for a particular gene of interest and just search for that, and it'll take us to our gene. So we can look across the whole genome, over 50 million variants in this case, or we can zero into a single gene. So that's quite a lot of sort of dynamic range when it comes to looking at data. But there are also interesting things we can do in terms of populations, and what we've tried to do on this Panoptes web browser, which was developed by a very brilliant team, Paul Voteran, Ben Jeffery, working with Alistair Miles and others, and Rachel Giacomo Antonio, who's in the audience here. And Rachel can tell you all about this. And what the team have tried to do is to put on the front page here a range of different features and buttons that allow you to quickly zero in to particular representations of the data, not just at the genomic level, but at the population level. So, for example, down here, there are different ways of representing how the population behaves in different regions of the genome. Now, one of the sort of curious features of large genomes, large recombining genomes of the sort that we have in mosquitoes, as opposed to many bacteria, is that we sort of don't reproduce clonally. We have this thing called sexual mating, and that thing results in a recombination. And one result of that very complex recombinational process, as it goes through multiple generations, is that you can end up with different parts of the genome having different ancestry. It's a very complex concept, but it's true, and we can see it in mosquitoes. So, for example, if we now do a neighbor joining tree of the mosquito population in the chromosome X telomeric region, and I've just got an example here, what we can see is that basically the mosquito population is assorting, rather like the parasite population I showed you earlier, and it's grouping by colour. In this case, colour of the dots around the outside mean country. So as with humans or with parasites, what's happening here is that in this part of the genome, different individuals look more similar to each other if they live near each other. But if we now go to another part of the genome, for example, this centromeric part of chromosome X, what we see is a very different picture. And here what's happening is that, again, to some first approximation, samples tend to cluster by their geographic location, but suddenly a group of samples splits away from all the rest. And what's happening here is a speciation process. It's the M and the S forms of Anopheles gambiae, Anopheles gambiae, what's now called Anopheles coluzi, a speciation process where in this parts of the genome, essentially, they're not sort of mating with each other. In this part of the genome, mosquitoes are behaving like different species. That's a sort of bizarre concept, isn't it? In part of the genome, you're looking like a species, and in another part of the genome, you're not looking like a species. And actually, what we're learning is that some of the rules of speciation are not quite as simple as we thought. That often there is quite a lot of mixing between species. But look at this other part of the genome here, which is the part of 2LA, which encodes the KDR insecticide resistance mutation. Here, all hell has broken loose, because what's happening here is there are multiple mutations. There are mutations causing KDR resistance, and they're zipping through the population, and they're breaking down geographic boundaries, and they're breaking through species boundaries. So one genome, but lots of different laws of spread of things. Now, again, the complexity of all this is vast, and turning this into simple epidemiological models of how resistance spreads and evolves, and how we turn that into decision support systems for malaria control and elimination is vastly complex. But then that's the world of big data, and that's what our job is, and that's what we're all going to be trying to do over the next few decades, trying to turn this type of complex big data into rational decision support systems. So it's an exciting time. Right, I think I must have had my time up by now. So let's zip through, and my closing slides are just to thank people and just to say, as you can imagine, I've just shown, shamelessly shown data from literally hundreds of researchers around the world, and I just want to name check a few of them. Firstly, our partners in the PFAS Community Project. I showed some data from that from around the world, and you can find these by going to this part of the Malaria Genomik website. And zooming in even further, this is just one group of partners, the track study that I talked about earlier, where there's investigators in different parts of Southeast Asia and in Africa who are working on detailed studies of artemisinin resistance. And then talking about our own team, it's a large team. I'm not going to mention everyone. I want to thank them all, but just to specifically mention Oliver Miotto, Roberto and Jacob, who were responsible for much of the PFAS population genetic analysis I mentioned, Will, Antoine and Mahir, who, as I mentioned, did the clone tree, did the mutational accumulation work I mentioned earlier. I've already mentioned Paul, Ben, Alistair and Rachel, who took a very strong lead in development of the Panoptes application. And thanks to all of you for your patience in listening to me.
So, do we have any questions for Dominic? At the back here. Can you wait for the speakers, please? There's the microphone.
Thank you for a great talk. I was wondering if you could comment more on what you see as the most promising methods going forward to really understand the mechanisms of genetic resistance generation, the population dynamics that are going on, for to really understand as quickly as possible the emergence of resistance, and what do you see as the most promising methods going forward there?
Well, it's a really massive question, and if I knew the answer, I wouldn't tell you, and I'd just go ahead and do it. But no, I mean, no, that's being facetious. I think there are some principles we can follow. I mean, I do genuinely think this is an area where we need to collect data, and we're in a position to collect data. I think that we are a little bit stuck in old-fashioned concepts. That it's terrifically expensive to do a type of mutation on a particular gene. Costs are driving down, and I think we should be doing a lot of sequencing as cheaply as possible, as systematically and prospective as possible and getting the data out there for people to look at. And I think one sort of slightly cliched comment I can make to you is I think this is the world of big data, and I think it's going to require quite a lot of statisticians, computer scientists, geneticists to look at it, just to look at lots of data, and I think there's lots in there. So I think part of it's about the community getting data there and examining it systematically in the ways that other big data problems are solved in computer science. I think the next thing is I think we just do have to have great phenotypic data and detailed phenotypic data and have particular use cases and scenarios. So I think for us, for example, in the area of insecticide resistance for mosquitoes, we really think it's dead important to do studies in a single village. I'm much more interested in having, prospective data on thousands of parasites, mosquitoes a year in a single village than I am in having it across the whole of the world. Understanding those fundamental ecological processes, how do, in a particular sort of larval breeding site, how many populations are there, how do they get on, what's their fate over the course of time. I think a third area, which is a little difficult to articulate, is the extraordinary advances that are going on statistically in how we translate genetic data into demographic inference. You may have heard about a paper that came out a couple of weeks ago called The Peopling of the British Isles. Has anyone heard that? Where basically, essentially by looking at genome-wide data in a bunch of people in the present day, we can make inferences about, how the British Isles were populated potentially 1,000 years ago, how Orkney is different from other islands out in the same region, how Cornwall is different from Devon. And we can use just the same sort of principles for mapping out demographic inference in a parasite population. So if we look at a county, a particular region where we're trying to eliminate malaria, we represent it all by pink blobs on a map. There's parasites there. Just imagine if we did human demography that way, and you were now trying to market a smartphone to blobs. I mean, you don't do market smartphones like you market smartphones by working out what different sorts of people live in different places and who they talk to, how they communicate. You need to understand those fundamental processes going on in human populations. And parasite populations are no different. So using genetic data to make very, very detailed demographic inferences about where parasites are moving from, rates of parasite migration, how many ethnic groups of parasite are there in the same village, is it a particular ethnic group spreading, how is that ethnic group related to other parasites living elsewhere, that will provide a deep understanding of the parasite population at the level of resolution that we understand human populations. So, and then I think finally, sorry, I've spoken too much to answer your question, but I do think then it's important to boil all this down to very specific use cases. What is happening explicitly at the borders of resistance between, say, northern Cambodia and northeastern Cambodia and Laos? What exactly is going on there? Getting detailed data, trying to make epidemiological models, and try to make genetic models, and see if we can match them up. So I think it's that last thing that's the most exciting. I'm sorry, that was a very long answer because your question was so complex.
We have one more question at the back.
Hi, thanks very much for your talk. Going back to that $10 million question in terms of, I don't want that 10 million, but the middle slide was interesting and it showed that little yellow dot of resistance. So you've gone through all the genetics, but what really are the environmental influences on resistance developing, on producing that yellow dot, whether it be on a village level or on a global level today, in terms of what are we doing wrong as humans with use of insecticides, with use of drugs and other things?
I think that is the basic question. I think that I think we have to start thinking organic. Fundamentally, we've got a finite number of resources, even if we get new drugs and vaccines, and we have to learn to use them better. And I think it is all about understanding how in different ecological settings, and the old malariologists were very clear about this, that getting rid of mosquitoes in Florida was different from getting rid of them in India or from Italy or wherever. Understanding those detailed ecological differences and how particular control regimes affect in different settings is important. And I think there's a very simple approach we can take, which is that now we can measure what's going on in the population, or we will be able to. You just simply watch what's going on with different interventions in different places and see which works best for controlling the disease without causing resistance in different places.
Two more questions and then we'll go.
Fantastic talk, thank you. Mine's one on policy. I know nothing about this area at all, but could you reassure me that in the process of research on malaria, where does the Gates Foundation come in? Are you talking with them? Are you bidding for their money? How does it kind of work?
Well, yes, okay. Well, we're very fortunate to have funding from the Gates Foundation, and I think they're very well aware of the sorts of issues we've been talking about. I think they're in the very difficult position of having to make very large decisions about resource allocation without the sort of knowledge about these types of things, like the previous question, what's going to happen if you do a particular intervention in a particular place? Are you going to make things better or worse? We don't have the information, so I think they're very keen to get that knowledge. I think that they have a particular challenge in connecting the sorts of genetic speak I've given you today with anything that relates to actionable knowledge in a national control program. And I think collectively we have to work together to bridge that gap by understanding how we translate this very, sophisticated genetics into very coherent epidemiological models with very simple decision support outputs.
When resistance spread to chloroquine and SB, it seems to have the same tools that we have today. But some of those resistant genes are still spreading, obviously, within Africa. I was wondering what you think might be learned from looking at the whole genome sequencing approach, studying that ongoing resistance spread, or looking at.
Again, I think you're absolutely spot on. This is a constantly, each of these things has a particular fitness advantage. It'll have a particular niche where it does well. There'll be other niches where it doesn't do well, and we have to live with them all. And I think those are still tools in our armamentarium. So understanding how we can play them off against each other, combine them, rotate them, and do fancy things to sort of force the parasite down a hole, as it were, is important. But I do agree with you. Getting detailed genetic data on those is just as important as the more fashionable new stuff.
So I'm going to call the first session to a close there. Thank our two speakers, David and Dominic, for a great